So jumping back into the second half of this chapter, what we're going to discuss today is the primary structure of proteins. We'll do a little bit of information around upper levels of structure, but we're going to save most of secondary, tertiary, quaternary for the next lecture. This lecture is going to be primary structure, some information about proteins in general, and some of the tools that we have available to us to isolate and purify proteins. We're not going to go into a lot of detail on sequencing because a lot of the sequencing technology that's in the text isn't the most current, but I encourage people to read about sequencing techniques, including the old ones and what's actually being used today, as well as some of the analytical techniques. And a lot of modern proteomics uses advanced mass spec as a tool, which is a little beyond the content that we really classically deliver in Chem 441. So there's obviously it's a large field and we're going to have to strategically omit some of the content, but I encourage people to pursue some of those other information. Obviously we're not going to be assessing on things that we don't cover in the text or in the lecture here. So without further ado, what are the essential questions for the chapter? And this one's pretty straightforward. What we're interested in is what structural form does a polypeptide chain assume, and how do we determine amino acid sequence, or more importantly, what is amino acid sequence, and what are the implications of amino acid sequence for the structure of the protein and in terms of its biological history. Also, what are some of the roles proteins play biologically? And I'm going to leave this slide up for just a second. Everyone can pause here and zip through here versus me just reading it aloud. And now we can get into it. So, what kind of structures do we see in proteins? So, we typically classify proteins based on their shapes and whether they're soluble or insoluble or what environment they tend to exist in. So the different shapes typically are globular. Let me grab a tool here. Are typically globular, fibrous, and membrane proteins. Globular proteins are exactly that. They exist structurally as semispherical proteins where the long strand is clapped in on itself, forming a spherical type structure. So myoglobin here is a really nice example of a globular protein. Fibrous proteins, collagen's a classic fibrous protein. It's a unique structure, which we'll discuss in further detail in the next chapter. But you have long fibrous structures. Typically you see these as part of structural elements or contractile elements in biological function. And then we've got membrane-bound proteins. And membrane proteins, like this one here, exist as part integrated into the membrane. So if we have a membrane, I'm just kind of doodling in the missing component here. The protein inserts into the membrane, and in this particular case it's a transmembrane protein, meaning it spans both sides, and it can provide feedback from one environment to another across the membrane, or can be used as a channel for controlling the environment internally or externally. So those are the three general types of shapes of proteins that we're usually interested or categorizing in. And then in terms of structure, we've got 4 levels of structure. And I've been using these terms a little bit, but it's good to formalize it now. So we have primary structure, which is entirely determined by sequence. We have secondary structure, which is the local structure formed immediately during the higher level of organization. And the 2 types that we're going to see overwhelmingly include alpha helices and beta strands or beta sheets. We also then have tertiary structure. So tertiary structure is the overall three-dimensional shape. So when we're discussing things in terms of something being fibrous or globular, that's going to be a discussion of tertiary structure. And then quaternary structure is the last level of organization where you are concerned about how multiple different peptide subunits with their own independent tertiary structures are associating with one another to form a quaternary structure. And what's important about quaternary structure is that as these units assemble, their association with one another drives refinement of the tertiary structures, which changes the overall structure function of those subunits, oftentimes activating them. So here's an example of primary structure. Primary structure is a literal sequence of amino acids. So if we were to approach from the amino terminal end, you can see lysine, glutamine, threonine, alanine, 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 lysine, phenylalanine, glutamine, arginine, etc. You can see that the primary structure doesn't change no matter how it's bound. If we were to disentangle this entire structure and just jumble it into a different structure, the primary organization of the amino acids wouldn't be altered. Now, in this particular structure, something that's worth to point out, and we discussed a little bit last time, is we have some disulfide bridges between cysteines, which provide refinement of how this sequence associates with itself. But that disulfide bridge isn't actually primary structure. That is going to be refinement of higher level structure. So most specifically tertiary. But that's all this is. It's just absolutely determined by amino acid sequence. So secondary structure. Secondary structure is defined by regular structures which extend along a single dimension. So the two that we typically pay attention to most frequently are the alpha helix and the beta strand. So let me grab color here. We've got our alpha helical structures and we've got beta strands. And what you typically see as abbreviations for these when you're looking at proteins, let me actually just jump up a few slides really fast. Like here you can see we have, and I'm messing things up, helical structures and more helical structures. And I'm not seeing any beta strands in these particular structures. But moving back, when you see this coiled system here, that is going to be shorthand for your alpha helix. You can also see them represented as cylinders, which we see here. And then we have the beta strand, which is a flat strand with an arrow, which gives you information about the direction of the protein's flow of primary sequence. So a little bit of information that we'll get out of these structures. The alpha helix coils around a central axis, which is what we're seeing here. And the R groups point outward as you move around the helix. In the beta strand, we talked about amide planes. The amide planes in this particular representation are perpendicular to the page. So we have these planar structures on edge, like this. And you see the R groups are being represented by the beta carbons, so these beta carbons are actually R groups. So the R groups alternatively point outward, giving you the least steric hindrance to these amide planes. Now let me just erase this really quick so we can focus. In addition to that, you'll see you have an alternation with the location of the oxygen, with this oxygen up, and then the oxygen down, and then the oxygen up, and then the oxygen down. So you have a really regular repeating pattern which gives you the most rigidity and the least steric hindrance possible. Now obviously there's a little bit of flexibility in these structures. These coils aren't absolutely rigid, and these strands aren't absolutely rigid. Let me see, hop back up again to that other slide. You see they're primarily rigid. If I look along this right here, for instance, pretty rigid structure. Pretty rigid structures. However, you do see a little bit of, let me see if I can clean this up a little bit for everybody so they can see what I'm doing. Grab a different color. You can see a little bit of distortion of the coil here. You see a little distortion of the coil here. And that's okay. A little distortion's alright. They are flexible. They're not super rigid. And that gives you the ability to tweak structure function of a protein. Tertiary structure is the structure of a protein in 3 dimensions. And there's lots of ways that we visualize these. You have space-filling models, you've got ribbon models, which I think are the most common. You occasionally just see backbone models, like in figure A here. But I think that B is going to be your most common model where you get the most useful information. And it's the one you see the most. Now this is a really nice representation because you can see right away from this that we have some helical structures, we've got some nice beta structures in yellow, and then we have some non-defined secondary structures with these green linkers, which are highly flexible. And then finally we have quaternary structure of protein. Quaternary structures, like I mentioned earlier, are multiple associated peptides, each of them having tertiary structure that is altered when they associate. And that assembly into quaternary structure refines the tertiary structure to allow for specific biological functions. So this is an example hemoglobin. It's a tetramer and it has 2 alpha units and 2 beta units. And we'll go into much more detail about hemoglobin once we've talked about allosteric regulation, because that's really the best place to discuss it. But this is a really nice example of quaternary structure. So protein's conformation can be described as its overall 3-dimensional structure. Now, because we're scientists and words have meaning, we have to be very particular about how we use words, especially when we're discussing things with other scientists so we don't seem silly. Case in point, conformation is the 3-dimensional orientation of a protein structure. Configuration is something totally different. Configuration involves the actual breaking of bonds if you're comparing configurations, and something that we used a lot of in organic chemistry. The conformation doesn't involve the actual breaking of bonds. It involves the spatial redistribution of something so that it reaches an energetic minimum or is activated by association with something else, but no bonds are actually broken in the altering of a conformation. So this takes me to a little bit of a segue, which I think is super, super important. Structure function. So a lot of people say it as one word. And the reason for that is because structure absolutely defines a protein's function. And we can trace that back all the way to primary structure, which is dictated by the sequence of amino acids which encode that sequence of, the DNA sequence which encodes the amino acid sequence which defines our primary structure. So, think about it this way. Structure of a protein determines its function. Now, we have the spectacularly specific chemical interactions which take place at the surface of the protein. And we have the core of the protein, which dictates how those surface chemistries align and refine them so that they are uniquely suited to interacting with substrates or with each other. But none of that supersedes the actual primary structure, because how a protein folds is ultimately defined by that primary structure. And this is why genetics has such a profound influence on the outcome of the biological role of proteins. And it's why point mutations sometimes can slightly alter or significantly alter or completely destroy the function of a protein, and why proteins can find diverse functions within biological systems with small modifications to their primary structure. So, we'll get into that quite a bit in the next chapter, but one of the questions that I nearly always ask is to explain why protein structure defines protein's function, and how they're inextricably linked. So, as we move through these chapters, keep that in mind. When I say protein structure function, I'm referring to not only modifications to the structure, but how that implicitly changes the function of the protein as well. So, a little bit of possibilities associated with conformational changes. Not all changes are equal in terms of their energetic preference, and that's because of the amide plane's rigidity. Because we have double bond character between the carbon and nitrogen, alpha carbon and nitrogen, we get this nice plane which only allows for rotation around specific pivot points. Now, in addition to having limited points of rotation, we also have side chains, bulky side chains, and we have oxygens, but these side chains are, since you know from the last chapter, substantially larger. Now, if we rotate these side chains into too close proximity to the oxygens or into other side chains, which would be over here, you can get all kinds of steric interactions which either reduce the energetic preference or actually fully inhibit the ability for that conformation to take place. So, even though we have quote-unquote free rotation around certain points in the protein, that doesn't mean that they're all energetically equally favored or likely to occur in a biological system. In fact, this amide plane significantly limits the number of possibilities that can occur under normal conditions. So, we're going to change directions a little bit and talk about the techniques that we use as part of working with proteins routinely. And it's important, even though the book doesn't emphasize this, to be aware of the tools of the trade. So, even though, like I said, in this particular text it's a bit of an afterthought, I think it's worth elaborating on quite a bit. So, isolation and purification of proteins. There's a variety of different techniques we can utilize, and usually when you're in the pursuit of a single protein or complement of proteins, you have to use a suite of different techniques which separate the protein based on various different chemical properties it's possessing. So some techniques are more suited towards bulk processing early on. Some are very specific and give you a lot of power once you've done some pre-processing, but aren't particularly effective right out the gate. So you have to also understand the context of when each technique is appropriate to use. And in terms of general categories or types of separations, there's solubility-based separations. And we typically call that salting in or salting out, changing the actual medium the protein's in so that it either stays in solution or crashes out of solution. You can separate proteins based on their relative ionic charges. So we have ion chromatography, electrophoresis, and isoelectric focusing, which use the various charged properties of proteins, often by manipulating their environment so that they can be separated from the other proteins around them. We can use polarity-based separation techniques. And these are chromatography, where you have a medium which has a specific polarity, and the proteins will associate to different degrees with that stationary phase. This is particularly useful in like C18 chromatography for liquid chromatography. This is more of a refined technique, and you see it more with LC. We have molecular size, where different proteins have different sizes and different shapes, and we can separate based on that using dialysis techniques. We can use gel electrophoresis. We can use ultracentrifugation as a technique, which separates proteins based on their sizes and shapes and ability to migrate through different mediums. And then we have a lot of power in specificity, specific binding characteristics. We have a protein which has a specific association for antibody or a small organic compound. You can attach those antibodies or small organic compounds to stationary media, which will help pull specific proteins out of solution. And obviously this is very powerful, but it takes a lot of work on the front end to accomplish it, unless it's a commonly done thing. You can purchase that stationary phase. And also, it typically benefits from a couple preliminary steps which clean up your solution first, so the specific binding points aren't overwhelmed with non-specific interactions from competing proteins. Let's go down this rabbit hole a little bit. So, another technique that we should be comfortable with is how protein concentrations are estimated. I talked about this a little bit, but it's worth pointing back to again. Obviously, an LC-MS type technique is your best for clear quantitation, but for semi-quantitative analysis or estimation, these are good techniques. So we can do colorimetric titrate, but we can also do colorimetric determination of proteins using these changes in copper's redox state, using often BCA proteins as a standard. And that's when you're doing benchtop colorimetric determinations, your most common technique. You can also use ELISA, which is an enzyme, let me point to it, enzyme-linked immunosorbent assay. Very powerful technique for quantifying specific proteins based on antibodies. And the way this technique works is you need a primary antibody, which is covalently attached to solid support. And then you do a wash where your protein is incubated in conjunction with this system, and the proteins strongly associate with the antibody. And then you gently wash everything else off, but not so strongly that the proteins dissociate from the antibody. Then you add a secondary antibody, which grabs the protein from another spot, and this typically has an enzyme attached to the secondary antibody, which interacts with a specific substrate, giving you a detectable product. And that detectable product can be light emitted in different frequencies. The enzyme itself can be fluorescent. There's a variety of modifications to the system used on the detection side that have various pros, cons, and degrees of power associated with them. But this is a very, very common technique. It's a very expensive technique because of the amount of specificity and the number of antibodies used. And so even though it's very powerful, you really need to know what you're doing when you do an ELISA. So solubility-based techniques are where you get your biggest bang for buck in doing protein isolations. Multiple groups on proteins determine its relative degree of solubility based on the concentration of its environment. And the environment can be altered to alter that protein solubility. And we can alter the environment by changing the degree of dissolved salts, the polarity of the solvent, which can cause denaturation. When something denatures, often it comes out of solution, the pH, and the temperature. Temperature often causes denaturation. So denaturation is the modification of the higher levels of structure of the protein. Doesn't change the primary structure, rarely changes the secondary structure, but modifies the higher levels of structure. And when that happens, often proteins will come out of solution. So quickly, in summary, why does altering a dissolved salt or polarity or pH or temperature change solubility? Well, dissolved salts interact with the surface of the protein, and if the protein surface is covered with those polar residues that interact with the aqueous environment, when you put those salts into solution, the salts are going to change the degree of interaction of those surface proteins with the aqueous system, or those surface residues with the aqueous system, and are going to alter the higher level of structure to where it can change it enough where it can come out of solution. Likewise, polarity of a solvent makes a big difference. If you start to modify the aqueous system with acetone, the internal residues of the protein, which are associated with each other in these hydrophobic systems, suddenly can creep outward and start interacting with the solvent, and that's going to turn a protein inside out to some extent, or completely alter its tertiary structure, and it can come crashing out of solution. pH can dramatically change a protein's structure, because as you change the pH, which we saw in the last lecture, you can be adding charges, both cationic or anionic charges, and that's going to significantly change the way a protein interacts with its environment. And finally, temperature. And that's an intuitive one for most people, because when we say cook an egg, you get a lot of denaturation, and by changing temperature, you're changing the way a protein is able to stably form its tertiary structures, and you can see it coming out of solution. So some of the ways, this is not something you guys need to memorize, but these are trends in the ways you see salts affecting the solubility of proteins. Different proteins are affected differentially by changing ionic strength. That's what we're seeing in this first slide, where you have ionic strength on the x-axis, and then you have the log adjustment of the solubility on the y-axis. And just by altering ionic strength, you can see that there's general trends with decreasing solubility as we increase ionic strength. However, there's a large variation in how soluble these proteins are to begin with, and at what ionic strength they come out of solution. So if we were to slowly increase ionic strength of all these proteins in solution, you would get them coming out of solution. All these would come together, at different ionic strengths. So you can see how powerful that would be as a tool for pulling those out of solution. Likewise, it's not just a matter of ionic strength, but how effective different salts are specifically as you increase ionic strength for changing the solubility of different proteins. This is a summary of how pH can affect solubility of globular proteins. And you can see there's a zone for each of these proteins where they have the most reduced solubility based on pH. And that typically, you have a window of reasonable function, and then as you leave that window, both above and below, solubility for these globular systems changes significantly. And that's why it's called salting in or salting out. There's a ton of different chromatographic techniques that we can use for isolating proteins. But all forms of chromatography are defined by the separation of different solutes based on their differential interactions between a stationary and a mobile phase. And I like to think of that as a bit of a tug of war between the two phases. So for those of you who haven't had quantitative analysis or aren't comfortable with chromatography as a general topic, what you have is, I'm going to blow this up, is a bead which is covered in, I'll say, we'll just call it C18. And this is a long carbon chain along the silicon bead. And so this makes this environment very hydrophobic. And then you have, these beads are obviously, you have a bunch of them. They're very, very small, 2-micron scale. And so it creates this sieving system where as a mobile phase with solutes flows through, things that are dissolved in the mobile phase interact with the surfaces that they pass by. And if the solute that's in that mobile phase has an affinity for the stationary phase, it'll spend time bound to that surface. Now, the trick to good chromatography is you always want to have some degree of affinity of a target solute for your stationary phase and some degree of affinity for the mobile phase. And what that does is it creates an equilibrium system where sometimes the solute is on the stationary phase and then it's in the mobile phase. Then it's on the stationary phase and then it's on the mobile phase. Stationary phase, mobile phase. And that specific ratio of time it spends between the two phases will determine how quickly it runs through a column. And that's the retention time. And so a particular solute's unique interaction or interactions between a stationary phase and a mobile phase will allow it to separate from any other solute that has a different ratio of interaction time between those two phases. Now this is just C18 chromatography, which is based on relative polarity, but there's a bunch of different types of stationary phases and mobile phases utilized in chromatography. And in protein separation, the ones you see the most common aren't polarity based, but you have ion exchange chromatography, we've got gel filtration chromatography, and we've got affinity chromatography. And we're going to talk about those three quite a bit. So ion exchange chromatography is probably the one, if you take biochemistry lab, you'll jump in and you'll do some ion exchange chromatography right out the gate. There's two types of ion exchange chromatography. You could probably guess what they are because there's two types of ions. There's cations and anions, positive and negatively charged. So we have specifically cation exchange chromatography, in which the resin, which makes up the stationary phase, is anionically charged and therefore has an affinity for cations that are in solution in the mobile phase. And then we have anion exchange chromatography, where the stationary phase, like DAOX or DEAE, is positively charged and therefore has an affinity for negatively charged residues for proteins that are in the mobile phase. And the way these work, this is a nice way to look at it. You load the sample mixture onto the column, which is full of whatever resin is appropriate for the separation that you're looking to do. And then you start to elute with, in this case, a low salt elution buffer. And the reason we're using low salt is we want the proteins to associate with the resin in their different characteristics. And they're going to run through at different speeds because they have different retention times. And some protein will come off quickly because it prefers the mobile phase. It has a relatively low affinity for the stationary phase. And then we'll have a second protein, that purple one, which has a greater retention factor because it has a higher affinity relative to the stationary phase, and it'll come off later. And then you can swap when you have something that's really holding on strong, and it would take a lot of time with this weaker solution. You can change your buffer to a higher salt concentration and flush remaining proteins that perhaps you're not interested in. Or there's lots of ways you can tune these systems. This is a stepwise elution where you have a low salt and a high salt, but with fancy systems with some automation, it's common for people to use what's called gradient elution, where these two, you'll have an A and a B, and you'll have two pumps which mix them, and the pump speeds will vary by program over the course of the elution progression. So you'll go from a very low salt strength gradually to a very high salt strength, and often this gives you a lot more separation power than you would get through these stepwise elutions, or a lot more efficiency. Gel filtration is another really slick technique. You'll also hear this referred to as size exclusion chromatography, and that's actually the term I was introduced to when I was using this technique. You also hear it referred to as molecular sieves. That's another term I've heard both size exclusion and molecular sieves used more commonly than gel filtration, but that's probably just the labs that I was at. All three are interchangeable. And what this particular technique does is it separates based on the size and the shape of the solute. So it's particularly useful for proteins because proteins often have not just different sizes, but unique shapes, which gives us another degree of power of separation. So the stationary phase in this particular case is a hydrated sponge-like material, which is porous. And that porosity is got a fairly defined window, so that large molecules are excluded from interacting with the surfaces of these beads, and smaller molecules get hung up in those pores in the beads and are retained. So just based on size, you'll see large molecules elute more quickly because they can't penetrate the surface. And here we have the blue, which are too big, and then you see the red and the red penetrate into the bead. And so the large ones go zipping through, and then the small ones are retained longer. And if we were to look at that in terms of elution, you'd have these nice Gaussian peaks. And you're not going to collect them. You're probably going to have some blue over here and some blue over here and some red over here and some red over here. It's almost never that clean as displayed, but you guys get the idea. This is a technique I use quite a bit. It's dialysis. The dialysis membrane has a defined porosity, a range of porosities, and you load your sample into the membrane, seal it up, put it into a low ionic strength solution on the other side, which is your solvent, and because of nature's tendency for things to try to reach equilibrium, two things will happen. One thing is that these solutes will try to escape so that you'll get an equilibrium with the internal concentration and the external concentration, and also you'll have an osmotic effect where the solvent will try to penetrate through here and cause some swelling, and that's an effect that also is trying to create an equilibrium between the concentrations internally and externally across the membrane. Now here's the trick. What you see in B is your outcome. If you have a membrane which has small enough pores, only the small solutes can make it out, and it retains all of the larger ones. So this is a really nice way to get rid of small contaminants, small molecular weight contaminants, and hold on to a purified protein. It's really common if you're salting out a protein to have a bunch of these large proteins, but then you also have a bunch of salt. And so one of the nice ways to desalt something following those initial steps is to do dialysis, because the salts can exit the membrane easily, but the membrane will retain all of your proteins. Unless it breaks catastrophically from swelling, which happens, and then you're sad. And then the last one that I want to go through is affinity chromatography. Affinity chromatography is an incredibly powerful technique, which, like ELISA, uses very specific protein-protein interactions or protein to small organic molecule interactions to isolate a protein based on its function or its affinity. And what you do is, just like we have all these stationary phases which can be coated with different polarity compounds, we can take a stationary phase and we can covalently attach either a metabolite, which is specific to a protein, or an antibody to a protein. And then as you elute a solution of proteins across that stationary phase, those proteins that have specificity for that antibody or that metabolite will grab hold. Everything else will run through with the past the stationary phase and elute, and then you can actually cause them to dissociate by slightly changing the structure of the protein using a little bit of a salt solution, and it'll crash those proteins off in a nice clean take. Like I said earlier, you nearly always need to do a series of isolation steps to get to a pure protein. If I ask a free response question, how would you go about isolating this protein? It's not uncommon for a student to say, well, I would just do immunity chromatography because it's so specific and I'm done. Well, the problem with that is if you go straight to the immunity chromatography step with a full complement of biological proteins from an extract, all those other proteins are going to interact non-specifically to some extent, and that's going to really cloud the effect, overwhelm those sites, so that you're really missing that specific interaction that makes it possible to get a nice clean separation with immunoaffinity chromatography. So you nearly always need to go from a crude extract to a bulk processing technique like a precipitation technique or ion exchange or size-based chromatography like molecular sieves, and then you can move on to an immunoaffinity chromatography technique. But oftentimes, you can get a pure protein without an immunoaffinity technique. So it really depends on the protein you're working with and what its attributes are. So other techniques we use in the analysis and visualization of protein include electrophoresis. And electrophoresis is the migration of anything that has ionic charge in an electric field. So we do it a lot with proteins, we do it with DNA, and we also do it with small organic compounds which carry charge because they're acidic or basic. In proteins, nearly all of the electrophoresis we do is gel electrophoresis. And you'll hear the term SDS page used a lot in labs. And that's an elaboration on the fact that the gel is polyacrylamide gel. So page is polyacrylamide gel electrophoresis. And SDS page is a modification on that technique where we are treating the proteins with sodium dodecyl sulfate, which denatures the proteins and removes the shape component of the electrophoresis through the gel and relies specifically on their mass. Modifications to this technique also include isoelectric focusing, where we have, which we'll talk about in a second, and two-dimensional electrophoresis, which combines isoelectric focusing with standard SDS page. There's also capillary variants, which are true to my heart, but that's in the spectrum of techniques not as widely utilized. So, polyacrylamide gel electrophoresis is page, and SDS page is a subtype of page which involves denaturation. And I don't want to go too far into this. Obviously, you guys don't need to remember the specifics of how acrylamide gels polymerize, but what you do need to know is that you can change the porosity of the gel based on the percent acrylamide you put into solution. High percent acrylamides are going to have gels which have smaller pores, and that means that the smaller proteins will migrate much more quickly through it, whereas larger proteins will be retained near the top. Now, here's the thing about that. You need to have an idea of what size protein you're looking at or trying to evaluate. And you might have to do a couple different percent acrylamide gels to tune the best visualization. So you'll run a variety of different gels, and your large proteins will be retained because they won't migrate as quickly, and then your small proteins will zip right through. Visualization of the proteins in the gel requires a couple different techniques. The most common is to use Kumasi blue, which gives you a visualization, but that only works for proteins that are really prevalent. There's other techniques which use Amino-based detection systems, which we're not going to get into right now. Western blotting is the one you've probably heard of the technique, but you actually transfer all of the proteins from the gel onto a blot, which is then washed with antibodies which have those fluorescent detection techniques or light emitting substrates associated with those enzymes or antibodies, and you get a much improved sensitivity and specificity with that technique. So back in the day, sure, we could do things with Kumasi blue because everybody was going after the easy low-hanging fruit, but now most of the proteins of interest are a little less prevalent, and Kumasi blue usually doesn't cut it. Here's what an actual SDS page looks like, where you can see the large kilodalton proteins retained more than the lighter kilodalton proteins. So isoelectric focusing is a really slick technique because if you're going to be using electrophoresis, your solute has to have a charge to migrate in the electric field. If you have a negative charge on a solute, it's going to run from the negative side towards the positive side, right? Well, proteins are able to be Zwitter ions at specific pHs, and that's unique to the proteins themselves. So there's going to be a pH where that protein is not net charged, and when you put that protein into that pH, it can't migrate in an electric field any longer. So the idea behind isoelectric focusing is you start with high pH in the gel, and it has a gradient which goes to a low pH, and this is then loaded into your electric field. You load your proteins on top, and then you apply the field, and as they migrate, as they hit that isoelectric point, they stop moving, so they don't run off the gel, and the point at which they stop moving corresponds to the pH at which they're no longer carrying a charge. So you're separating in one dimension based on their relative pH at which they are not charged. The really cool thing about this is then you can turn this on its side and run it through a classic gel. And this is two-dimensional gel electrophoresis. So you can isoelectrofocus in one dimension, turn it on its side, and then run the proteins out based just on their size and shape. And you get these really high resolving power where each of these dots on this gel have been isoelectrofocused in this plane and then run out based on size and shape in the other plane. And then you can go in, if you do an antibody stain, that gives you exactly what you need, and you can cut out something specific from the gel, and voila, you have a highly purified protein. The downside of this technique is it's not highly reproducible, so it takes a little bit of art to it. So now that we've talked about all of these tools, let's kind of wrap it up with going back to amino acid sequence and the information we get out about that, get from that. Like I mentioned earlier, and I want to keep saying it over and over so that it really hammers it home, the sequence, the primary structure of a protein, determines its function. If you alter the sequence, you will alter the way the protein folds, either a little bit or a lot, and that's going to change the way it functions. You're going to see differences in the amino acid compositions of different categories of proteins. For instance, membrane proteins have a lot more hydrophobic residues because the internal component of that protein is strongly associated with the lipid environment, whereas fibrous proteins often have unique atypical sequences, with collagen being a really good example. Now, there's often homology in proteins between organisms. And we refer to some proteins as highly conserved and some proteins as highly variable. And proteins which are associated with some of the cores of metabolism are often highly, highly conserved, which is why we can use model organisms such as zebrafish or even nematodes to study ourselves in ways that are inconvenient or unethical to study ourselves. A good example of this is cytochrome C. Cytochrome C is highly conserved, and its function is extraordinarily refined. and we'll be going down a discussion specific to cytochrome C in the future. But how a protein has been modified over time is also traceable back to its primary sequence. And how a primary sequence changes over time is then passed on through the genetic code from organism to organism to organism. And this is the mechanism by which we can really evaluate how our genetic history has been traced from one organism to another, or how one organism's genetic information historically became the basis for many organisms down the road. And this is just a quick description of the relative composition frequency that you'll see amino acids in different proteins. And you'll see that leucine and alanine, for instance, occur very frequently, but a lot of the tryptophan, cysteines, much less frequently. So all amino acids don't occur in the same frequencies. This is for all proteins in distribution. Some proteins will have substantially more cysteine or tryptophan than others. They'll have a lot more aromatic residues in general with these black bars, which are less common. Then we have, fairly low frequency of sulfur-containing amino acids. Now, going back to alignment, because computing power is such an amazing tool for genetics and proteomics, we are able to rapidly find alignment of sequences in primary sequence. And this gives us an ability to actually quantify the homology between proteins. So in here we can see all these points where we see homology in sequence. And then we see zones where we have differences. You can see gaps where something was inserted here, creating a longer sequence. And this gives us insight into, for instance, if we were to evaluate this change would continue to be passed on in subsequent versions of the organism, whereas here, if we were to not have it, we would have information about when it was gained or lost. And this is really how we develop these genetic trees, which give us information about how things are related at the most fundamental level. So I think it's important, it's responsible, when people talk about evolution being a theory. Just like gravity is a theory, but evolution's a theory. It's important to know, as somebody who's been exposed to at least 400 level biochemistry, and hopefully beyond that, that if you're looking for evidence of evolution, our entire genetic code and the conservation of primary and secondary and tertiary structures in proteins, all of the data we have from genetics and proteomics across all organisms is a body of data which fundamentally supports evolution. We have a spectacular body of evidence of support of evolution because of the way we code for proteins at the genetic level and how we transmit that genetic information from organism to organism to organism over time. And through statistical evaluations of homology, we have spectacular insight into how organisms have branched over time, and how sometimes those branching incidences have coincided with historical geological events, or other environmental stressors. So I'm just going to leave that with everybody. to think through. But the next somebody says, oh, you think we came from monkeys? There's a whole lot more that we can say that typically isn't said. Oh, and this is just discussing proteins with common evolutionary origin, which I think we just did justice to. And this is another one, same example, and these are just globin chains comparing human hemoglobin and myoglobin, just to show that despite having both of these proteins in ourselves, that there's clear homology within them, which means that at one point we had a common protein which got multiple uses and then specified over time with point mutations and insertions and deletions. But at the core, we have an enormous amount of overlap for what they are and where they came from. So since we're talking about homology, a couple points. Apparently different proteins often share common ancestry. So in this particular case, you have a human protein, lactalbumin, and you have an egg protein from egg whites, lysozyme. And if you look at these, they have obvious homology. There's a lot of sequence homology, and there's a lot of homology in their secondary and tertiary structures. There's absolutely no relationship in terms of their biological function, because this protein plays a very different role biologically than this protein. But that's not uncommon. It's not uncommon for proteins which share sequence homology and share structural homology to find different niches biologically. Some people will argue my assertion that structure equals function because of this. Some people will say, well, structure doesn't equal function because function can change so rapidly with the same structures. But I think that's missing the forest for the trees, because what we see here are still very specific biological functions being dictated by a structure. If you were to significantly alter the structure, the function of either of these proteins would be altered. So I'm still going to stand by structure. Function is the same thing. Another really good example of this is snake venom, or venom in general, where the salivary glands become highly specialized in certain animals to deliver a suite of different proteins. And some of those proteins are digestive, some of them are hemolytic, and it turns out when you were to inject them as part of a bite, it increases the lethality of that bite, so that particular protein expressed in the salivary glands is retained, and then you get competitive advantage, so it's passed on and passed on and passed on. And as other mutations take place in those proteins, they become even more adept. That's one of my favorite examples of how a protein which has had one function can become incredibly powerful with a different function, with just a little jump of how it's being applied in the organism. Another example is how you can have very similar tertiary structure, although you have a totally different sequence with very little sequence homology, and have different function because of that. This is a really interesting example of hexokinase compared to G-actin. And if you look at these, you see a lot of homology with the tertiary structure. You see a lot of homology with how the secondary structures are arranged, but there's very limited homology at the primary level, and the functions are totally different. There's often multiple ways to skin cats biologically, and evolution provides an interesting mechanism by which we can reach the same outcome through different paths. And this is called convergent evolution. And it's worth noting that because we can form helical structures through multiple sequences, and we can form beta sheets, you get these regular repeating patterns through a variety of different tools, and then they can assemble in only so many ways. So, in the end, even though primary sequence will still dictate function, we can get to that same function through a variety of different mechanisms. Last couple slides, I think it's important to mention that there's all kinds of modifications to proteins that involve things other than amino acids. Proteins can be modified via phosphorylation, acetylation, methylation, acylation, and adenylation, and a variety of other tools. And all of these modifications change a protein's structure function and are often mechanisms by which biological systems exert control over a protein's function, turn like an on-off button or tool which varies the degree to how on or off a protein is. And lastly, what are the many biological functions of proteins? I thought about putting the slide at the very beginning, but I actually think it's just a nice way to wrap up the lecture. To summarize the diversity of biological functions proteins serve. Proteins can serve as enzymes, which have catalytic function, which are highly specific and essential for our metabolism. They can be regulatory in terms of controlling metabolic or gene expression functions. They can be DNA binding. They can associate in conjunction with RNA as part of our gene regulatory processes. They have frequently exert transport functions, shuttling functions, which give you spatial control within cellular events. They can have storage functions. They can cause physical motion through contraction in our muscles. They have structural roles with collagen. They are involved in our signaling pathways, which help regulate our whole organism responses to our outward environments as well as our immune function. There's an elaboration from the immune function. They have protective roles. We have signaling proteins that not just signal between cells, but are exerted through hormones. There's so many roles proteins play biologically. They're just spectacularly diverse. So that's it for this lecture. We're going to pick up the next lecture on higher level protein structure function in sometime this next week. So I'll have that up for everybody in the not too distant future.